everyone. It is my pleasure to introduce today's speaker, Sergio Muñoz-Gómez. Sergio is joining us from Dalhousie University from Nova Scotia, Halifax. And he's a graduate student in the cell biology and evolutionary biology, as well as bioinformatics departments of Dalhousie University. Sergio received his bachelor's degree in biology from University of Antigua in Colombia, and then he moved to Canada to receive his PhD, which he's still working towards. But he received a Master's of Science degree in 2015 from Dalhousie University, and he stayed further in furtherance of his research. I met Sergio during my visit to Dalhousie University a few weeks ago. And I was very impressed by his vision, his perspective on questions that really require a big vision, but yet he follows a very detailed molecular approach in order to answer the big questions in science. And I thought that his research would be very relevant for the astrobiology community. And I look forward to listening Sergio and see hear about what he will talk to us today. So thank you, Sergio, and I will mute myself and it's all yours. Thanks.
Okay, thank you very much, Betul, and thank you for the invitation. I just want to make sure that you are looking at my slide right now. Is that right?
No, I see a black screen. Is it just me?
I see a black screen also.
I see a mouse
screen.
I see a mouse.
Let me try to fix this.
There you go.
Now?
Yep, that's good.
Is it working now?
Yep.
Okay, so I want to talk today about what I'm focusing for my PhD project, which is the early evolution of mitochondria. And more specifically, I want to talk about how mitochondria became very specialized respiratory or bioenergetic organelles of eukaryotes, which is one of my main interests. So I just want to start by saying something very general, something that probably most of you know, which is that mitochondria are organelles of endosymbiotic origin, which means that unlike all the major organelles of the eukaryotic cell, such as the nucleus, the endomembrane system, the ER, the Golgi apparatus, the flagella, and the cytoskeleton, it turns out that mitochondria actually came from outside the cell. That is, mitochondria have exogenous or endosymbiotic origin. So in our very distant past, a host, probably some kind of archaeon, we now have found a very close relative of eukaryotes, which is called Lochiarchaeum, probably internalized the ancestor of mitochondria. So that means that mitochondria have an endosymbiotic origin. So it didn't evolve from within the cell, but it came from outside. So we know that mitochondria came from outside the cell, but we don't know exactly what kind of cell gave rise to mitochondria. So in the last 2 or 3 decades, several different phylogenetic studies have actually revealed the major group from which mitochondria evolved. And it turns out that the group that gave rise to mitochondria is called alpha bacteria. And here on the screen, we have a very simplified phylogenetic tree of this group, alpha bacteria. And it turns out that when we incorporate mitochondrial sequences into this analysis, mitochondria branch at the base of alpha bacteria, but we are not certain whether it is branching with all this major group up here or with the other group that is called rickettsialis. So this larger group that is composed of 5 different orders is usually composed of very versatile and metabolically complex bacteria. But the other group that is called the rickettsialis is composed by very simplified and intracellular bacteria. So depending on where mitochondria is branching, which is something we don't know yet, that position will have very different implications for the origin of mitochondria and therefore also eukaryotes. So under the first alternative, then the ancestor of mitochondria would have been very probably a purple non-sulfur bacterium, which is an alpha proteobacterium that had the capability of performing photosynthesis, an oxygenic photosynthesis. But under the other alternative, in which mitochondria evolved from Rickettsialis, or has a common ancestor with Rickettsialis, then the ancestor of mitochondria was probably already a very reduced bacterium that had adapted to a specific intracellular lifestyle. So then that bacterium would have been an intracellular endosymbiont. So we have two different options that have different implications for the very early evolution of mitochondria. Now, uh, the origin of mitochondria from a specific bacterium and its conversion into an organelle require a lot of changes. So irrespective of whether the ancestor of mitochondria was a purple bacterium, a complex bacterium, or a very simplified endosymbiotic bacterium, a lot of very different changes happen to give rise to modern mitochondria, which are very integrated organelles in the economy of the eukaryotic cell. So because so many changes happen, and we know what kinds of changes happen, but we don't know in what order and when they happened. So many things happen that I call these the dark ages of mitochondrial evolution because we don't know in what order and exactly how they happen, although we know already what happened. So just to clarify some terminology, when I talk about the pre-mitochondria, I'm referring to the alpha proteobacterial ancestors of mitochondria, whether free-living or endosymbiotic. And then when I talk about mitochondria, I'm talking about a fully developed organelle uh, that is present in modern eukaryotes. And the transition between the pre-mitochondria and a fully developed organelle um, happen throughout many stages that I designate the proto-mitochondria. So just to make this point that a lot of changes had to happen between the bacterial ancestor of mitochondria and fully developed mitochondria, I want to show you this slide in which basically we had to go from a proteome that was basically a proteome is the whole set of proteins of an organism. So a proteome that was mostly composed of alpha proteobacterial proteins to a proteome in more mitochondria, which is 80% eukaryotic and only 20% alpha proteobacteria. And what this means is that uh, first, that the ancestral alpha proteobacterial proteome was highly reduced. A lot of proteins were lost or transferred to the eukaryotic nucleus. And then the second major point that I want to make is that a significant fraction of the mitochondrial proteome was actually invented by the eukaryotic host. So this is just to make the point that more mitochondria, even though they descend from alpha proteobacteria, they're drastically different in many different aspects. And this is a list of some of the major innovations that had to happen in order to transform a bacterium into the mitochondrial organelle. So first we had, for example, some gene loss. A lot of genes had to be lost in adaptation to an intracellular lifestyle, and therefore the genome of the bacterium was reduced. But then we also have some gene transfer to the nucleus. Some of these genes were not lost, but were transferred to the nucleus of the host. And then the host started exerting certain control on the biogenesis and the development of the new endosymbiont or organelle. And then because genes were already in the nucleus of a host, those proteins were being synthesized in cytosolic ribosomes. And those proteins had to come back to the nascent organelle somehow. So protein targeting or import machinery had to evolve as well. And this is probably one of the most interesting aspects of mitochondrial evolution because the protein import machinery actually has its origins in the protein export machinery of alpha proteobacteria. What happened was that this machinery was modified with the help of new eukaryotic proteins in order to import proteins rather than secrete proteins. Other changes that happen during the transition to the origin of mitochondria include the origin of a mechanism to divide mitochondria, which is fully controlled by the host. Also, interactions with the cytoskeleton and different endomembranes in order to shape mitochondria, distribute mitochondria, and inherit mitochondria in different ways. So the host had to gain control over the newly evolving organelle by basically uh, making them interact with the cytoskeleton and position those organelles in key places so the host could maximize the bioenergetic output of the new organelle. Then it turns out that some mitochondria can actually fuse, and this is also an adaptation that some eukaryotes have found to maximize uh, respiratory efficiency. And finally, the development of cristae, which are invaginations of the mitochondrial inner membrane, was another very important step in the specialization of mitochondria as a respiratory organelle. But mitochondria are more than the powerhouses of the eukaryotic cell. And this is just to show that mitochondria are very, very integrated into the economy of the host, of the eukaryotic host, and therefore they have also acquired new functions. So in addition to aerobic respiration and accessory functions such as the biosynthesis of ubiquinol, cardiolipin, and heme, mitochondria are also involved in the metabolism of amino acids and fatty acids, and also in the biogenesis of iron-sulfur clusters, which are cofactors of proteins are actually exported from the mitochondria to the cytoplasm. So cytoplasmic proteins can also make use of these cofactors that are only synthesized inside the mitochondria. And additionally, in some multicellular organisms in particular, mitochondria have actually acquired very important roles in cell signaling and apoptosis. So now I want to talk about how the mitochondrion specialized as a bioenergetic organelle. So what the thing is that there were two major sets of adaptations that enable mitochondria to be a very, very efficient bioenergetic organelle of eukaryotes. Because if you think about the bacterial ancestors of mitochondria, they have a lot of functions, but modern mitochondria are very specialized to very few functions, among which respiration is probably the most important. So a lot of these two major adaptations allowed mitochondria to perform these few functions very efficiently. So the first one is related to the overall morphology of mitochondria and the distribution of mitochondria. And therefore, this means that mitochondrial dynamic behavior is an adaptation to bioenergetic function. So if we look at the distribution and the shape of mitochondria in very different eukaryotes, we see that, well, precisely mitochondria assume shapes and localizations um, that maximize their function. For example, in sperm cells, mitochondria actually localize to the neck of these cells, where a lot of ATP is consumed for flagellar movement. In E cells, for example, mitochondria fuse to form networks that localize to the periphery of the cell, and therefore is close to oxygen, and also this particular morphology apparently is contributing to maximize respiratory output, mostly because products are shared between different mitochondria, and also mitochondrial DNA can be repaired by homologous recombination when we fuse these mitochondria. And in ciliates, which are like very complex cells, very metabolically active complex cells, we see a lot of mitochondria, and all these mitochondria are localized to precisely the parts of the cell that need more ATP. But I don't want to talk about the first set of adaptations today. I'm going to focus on the second major set of adaptations, which have to do with mitochondrial ultrastructure. Because, well, right now during my PhD, this is the major focus of my studies. So it turns out that mitochondrial bioenergetics also depends on mitochondrial ultrastructure. So if we have a closer look at mitochondrial ultrastructure, more specifically the envelope of mitochondria, we see that, well, mitochondria is surrounded by two different membranes, an outer membrane and an inner membrane. And the inner membrane itself can be subdivided into two different domains, the inner boundary membrane and the crystal membrane. And each one of those different subdomains have different functions. Um, For example, the inner boundary membrane is mostly in charge of importing proteins and transporting metabolites between the mitochondrial matrix and the cytoplasm, whereas the crystal membranes are those membranes that protrude into the mitochondrial matrix and allocate all the machinery for oxidative phosphorylation or aerobic respiration. So these membranes are enriched in protein complexes that produce energy. Now, these two different membrane subdomains are connected to each other as specific tubular or neck-like structures called crystal junctions. And well, and crystal junctions are very important because it is these tubular structures that connect those different membrane subdomains that are creating crystal microcompartments, and therefore by creating microcompartments, these crystal junctions are optimizing respiration by concentrating solids and all these reactions in cristae. And it turns out there's a protein complex called micose that is responsible for creating crystal junctions. So micose constitutes the molecular basis of mitochondrial cristae. And micose is a protein complex that is composed of 6 different proteins. And these proteins all localize to crystal junctions, and they determine the development of cristae. So we have 6 different proteins, of which MYC60 and MYC10 are the two most important and central components of the protein complex. So what happens when we delete micose from E cells, for example, is that we go from a normal-looking mitochondrial with beautiful cristae and beautiful crystal junctions to mitochondria that that lose the crystal junctions. And because mitochondria lose crystal junctions because of the absence of micose, then we see an internal accumulation of membrane vesicles. Basically, what's going on is that because crystal junctions are being lost, then cristae are detaching from the inner boundary membrane, and crystal membranes are accumulating in the matrix of mitochondria. And this obviously has a very deleterious effect in the respiratory function of mitochondria. So it turns out that micose, this protein complex, is combining different functions in order to create crystal junctions. So the first function of micose, mostly through a subunit called MYC60, is to anchor the mitochondrial inner membrane to the mitochondrial outer membrane, which is a very important function to stabilize mitochondrial cristae. The second function of micose is to create negative curvature, so the inner membrane can actually invaginate and give rise to crystal membranes. And the protein and the micose subunit that is in charge of this is called MYC10. And then there's a third function that we can attribute to micose, which is the differentiation of this of the inner boundary membrane from the crystal membrane. And there are probably 3-4 accessory proteins or stabilizing factors are in charge of this. And one of these two proteins that are probably doing this are MYC26 and MYC27, which can actually bind cardiolipin, which is a very important lipid of the mitochondrial inner membrane. And by doing this, micose is segregating the lipid composition of the crystal membrane from the inner boundary membrane. So as part of my PhD project, I'm studying the evolution of cristae, or the evolution of mitochondrial structure. So the first thing that I did was to investigate the evolutionary history of micos. I wanted to see how micos had evolved and if that could give me a clue of how mitochondrial cristae, and therefore how mitochondria as a whole, um, enable the specialization of mitochondria as a respiratory organelle. So I did BLAST and HMM searches against eukaryote genomes and transcriptomes. Basically, as I assembled a data set of very diverse eukaryotes that represented the whole eukaryotic diversity. And then I used some reference proteins, and I searched these proteins against the genomes of all these eukaryotes. And the first conclusion that I got after analyzing my results was that both micos and therefore crystal junctions are ancestral features of eukaryotes. So in animal and in fungi, micos is composed of 6 different subunits. And what I found was that of these different 6 subunits, at least 3 subunits are consistently found in all the major groups of eukaryotes, which allow us eventually to infer that the last common ancestor of all eukaryotes already had a micos complex composed of at least 3 different subunits that was controlling the development of mitochondrial cristae. So what this means is that the last common ancestor of all modern mitochondria already had the capability of developing cristae. So the second major finding that I did was that micos co-occurs with mitochondrial cristae. So it turns out that there are a lot of eukaryotes that have adapted to an anaerobic lifestyle, and by doing so, they have lost the capability of aerobic respiration. So in those eukaryotes that perform aerobic respiration and therefore have cristae, we find a typical micos complex. And even in those eukaryotes that are sometimes anaerobic but still have cristae, we see that they still have retained the 2 most important subunits of micos, MYC60 and MYC10, and therefore they have the capability of developing fully functional cristae. But there are some instances in which there are certain anaerobic mitochondria that don't have cristae per se, but have certain convoluted morphology of the inner mitochondrial membrane. And such example is Cryptosporidium parvum, which is a parasite that has adapted to very hypoxic environments. And this organism does not perform aerobic respiration and does not have cristae in its mitochondrion, but it has conserved the MYC10 subunit, and the presence of this subunit in its genome seems to be explaining the very convoluted morphology of the inner mitochondrial membrane. But then, more interestingly, in those organisms that have more derived mitochondria that are called hydrogenosomes or mitosomes, and therefore that don't perform aerobic respiration, we see no trace of any micos subunit in their genomes. So the overall picture here is that the presence of the micos complex correlates with the presence of cristae. Once you lose the micos complex, you're basically losing the capability of developing fully functional cristae. Of course, you also need to lose the machinery that you allocate in these membranes. For example, the oxidative phosphorylation machinery. So once you lose those 2 things, you are not capable of developing mitochondrial cristae anymore. And the third major finding was that it turns out the micos is present in alpha bacteria. And if you remember well, alpha bacteria is the group from which mitochondria evolved. But even within alpha bacteria, micos is not present in all of them. Micos is only restricted to certain groups of alpha bacteria, which are those proteobacteria that are more metabolically complex. So we don't see mycos or MYC60, which is the major subunit of mycos, in the Rickettsialis or in the third 11 group. And if we have a closer look at the homologue of MYC60 in alpha proteobacteria, we see that this protein actually has the same overall structure. So in eukaryotes, MYC60 has a targeting signal or a presequence that is in charge of targeting the protein to the mitochondrion. But then we have a transmembrane segment that is responsible for anchoring the protein MYC60 to the inner membrane. Then that is followed by a coil region, and finally followed by a conserved myofilament domain that is probably likely the most important part of the protein because it is highly conserved at the sequence level. So in terms of conservation, the whole protein is conserved at the structural level, but only the C-terminal part of the protein, the part in green, is conserved at the sequence level. And this is actually the part of the protein that allowed me to detect this protein in alpha proteobacteria. So now, it turns out that mycos has alpha proteobacterial origin. And also the structure of mycos in both eukaryotes and alpha proteobacteria is very similar. So this raises the question of whether there has been a functional conservation between alpha proteobacterial and mitochondrial MYC60. Again, MYC60 is the main component and central component of mycos, and it's probably largely responsible for its function. So in order to answer this question, it is interesting to consider the observation that it turns out that a lot of those alpha proteobacteria develop intracytoplasmic membranes or ICMs that resemble mitochondrial cristae. So we have two examples here. We have Rhodospirillum rubrum and Rhodopseudomonas palustris, which are two different alpha proteobacteria that photosynthesize, and they allocate the photosynthetic machinery to these membranes in order to produce bioenergy. So in this regard, ICMs in bacteria are very similar to mitochondrial cristae because they are responsible for bioenergetic function. So basically, they produce a lot of ATP by housing protein complexes that transfer electrons and that pump protons and eventually do phosphorylation. And even within alpha proteobacteria, ICMs are very diverse morphologically and functionally, but they all perform bioenergetic functions. So the ancestral type of ICMs in alpha proteobacteria is that is capable of performing an oxygenic photosynthesis. But it turns out that some alpha proteobacteria have transformed these ICMs to carry out different physiological processes. For example, some alpha proteobacteria use these membranes for nitrification. Other alpha proteobacteria use them for methane oxidation. And still, I argue that other alpha proteobacteria have co-opted these membranes for magnetotaxis, which is basically a capability that certain bacteria have to navigate the magnetic field of the Earth and position themselves within this magnetic field in order to reach their optimal environment. So what I want to talk about now is the homology hypothesis. Basically, everything I've said is with the aim of trying to connect mitochondrial cristae with alpha proteobacterial ICMs, and this is the homology hypothesis. The homology hypothesis basically states that mitochondrial cristae, which are these very important adaptations for bioenergetic function, evolved from alpha proteobacterial ICMs. And if this is true, then I would expect that MYC60, which is the main component of mycos, is performing the same role in alpha proteobacterial envelopes as eukaryotic MYC60 is doing in mitochondria. So we know what MYC60 is doing in mitochondria, but we don't know what MYC60 is doing in alpha proteobacteria. But given that alpha proteobacteria also use these membranes for bioenergetic functions, then I would hypothesize that MYC60 might be performing the same role. And if this is true, then that would be supporting the homology hypothesis between these two different structures. And the homology hypothesis is not new. It is not my idea. It actually traces back to the 80s, where different microscopists had actually discovered this similarity between alpha proteobacteria and mitochondria. And now the homology hypothesis relies on another hypothesis that I call the functional conservation hypothesis for MYC60. And there are actually several lines of evidence that argue in favor of the functional conservation hypothesis and therefore also the homology hypothesis between alpha proteobacterial ICMs and mitochondrial cristae. So the first line of evidence is the sequence similarity that there is between the two versions of MYC60. The second line of evidence is that both alpha proteobacterial and mitochondrial MYC60 have the same structure in general. So despite more than 2 billion years of evolution, the structure of the protein has remained the same. So that is actually evidence for the functional conservation of this protein. Then there are two more important lines of evidence. The third one is that it turns out that MYC60 in alpha proteobacteria is expressed precisely under those conditions in which ICMs develop. So, for example, several alpha proteobacterial transition between a heterotrophic and a photosynthetic growth. And when these alpha proteobacterial are transitioning to a photosynthetic growth, they start developing ICMs. And this development of ICMs coincides with the expression profile of MYC60 in these bacteria. And moreover, when I was looking through the literature, I found at least 3 or 4 studies in which they had analyzed the protein composition of isolated ICMs. And it turns out that MYC60, this protein, is actually enriched in these ICMs. So when I take together all these lines of evidence, I gain certain confidence in that MYC60 is probably doing the same thing in the two groups, in alpha proteobacteria and in mitochondria, which is further support for the homology hypothesis that the two structures are related and that cristae evolved from ICMs. So now we know that mitochondria are, well, we know what MYC60 is doing in mitochondria, but we don't know what MYC60 is doing in alpha proteobacteria. So the question is, what are the possible mechanistic roles of MYC60 in alpha proteobacteria? So here we have an alpha proteobacterial envelope, and basically by inference from what we know from MYC60 mitochondria, I would argue that MYC60 might be creating ICM junctions basically by undergoing homotypic interactions between different MYC60 subunits. MYC60 would be creating these necks that compartmentalize ICM metabolism. But then we know very well that in mitochondria, MYC60 is also involved in creating contact sites between the inner membrane and the outer membrane. Now, alpha proteobacteria also have two different membranes, and they have the same protein partners that MYC60 have in the outer membrane. So I argue that MYC60 is probably creating these contact sites between the two envelopes. And this would be an additional function to stabilize ICMs in alpha proteobacteria. And by doing these two things, 1 and 2, then MYC60 would probably also compartmentalize ICMs by restricting metabolites and different proteins into the ICM lumen, and therefore improving the efficiency of the reactions that are happening there. An additional function of MIG60, given that, well, the protein composition of the ICM is clearly different from the protein composition of the cytoplasmic membrane of alpha proteobacteria, is that MIG60 would be involved in differentiating or somehow restricting the diffusion of protein complexes between the ICM and the cytoplasmic membrane of alpha proteobacteria. And finally, I think that if it turns out that MIG60 is not actually involved in creating ICM junctions or contact sites, then it is probably assisting protein secretion by basically bringing together proteins of the cytoplasmic membrane and the outer membrane. So what I'm doing right now in the lab is to test the homology hypothesis. So I'm currently working on 2 different alpha proteobacteria, and I'm knocking out, well, I knocked out the genes, 2 different genes, MIG60 and another candidate protein that I have, in order to see how the morphology of ICMs is disrupted. What I expect from this is to see that the ICMs are actually separating from the alpha proteobacterial envelope. Something else that I'm doing right now is that I have made the strains that will overexpress these proteins to see what the effect of the overexpression of these proteins is on these bacterial cells. I predict that the overexpression of these proteins are going to increase the connections between the ICMs and the envelope, or between different ICMs. And another thing that I'm doing in order to experimentally test the homology hypothesis or my bioinformatic predictions is that I'm currently working with a yeast knockout strain that does not have the MIG60 homologue, and therefore has those mitochondria that accumulate internal crystal membranes. But I'm introducing the bacterial homologues of these proteins into yeast to see whether their expression is going to rescue the original phenotype or not. So if it turns out that the bacterial homologues, even though they're very distant, rescue the phenotype of the yeast mutant, then that would be evidence for the conservation hypothesis, the functional conservation hypothesis between the 2 versions of the protein, and also the homology hypothesis that basically states that cristae, respiratory cristae mitochondria, evolved from ICMs. Now, the second part of my PhD, which I also started already, is related to investigating the phylogenetic origin of mitochondria. So in order to do this, I started the sequencing of some new alpha proteobacteria that branch very, very deeply in the alpha proteobacterial tree. And I hope that by increasing the sampling of these different diverse alpha proteobacteria, I will have more resolution in order to address the question of where the mitochondrial lineage branches within proteobacteria. And if I'm able to add some resolution to the phylogenetic tree, then we will be able to have a better idea of how the ancestor of mitochondria looked like. So in this regard, I'm going to leave you with this question of what was the nature of the mitochondrial ancestor. We basically have 2 options. One is that the ancestor that gave rise to mitochondria was a very specialized and highly reduced alpha proteobacterium, like a parasitic rickettsial, or 2, that the ancestor of mitochondria was a more complex and metabolically versatile alpha proteobacterium, probably a stem alpha proteobacterium that have the capability of performing an oxygenic photosynthesis. I myself tend to think that it was probably the second option, in which the ancestor of mitochondria was a more complex bacterium that probably provided a stronger selective advantage to the host. And if you think about what I've told you so far, basically my investigation is pointing towards the direction that the ancestor of mitochondria had the capability of performing ICMs, but Rickettsialis or very reduced alpha puribacterium cannot develop ICMs. So that's why I think that probably the ancestor of mitochondria was a more complex bacterium that used ICMs for photosynthesis, and that during the evolution of mitochondria, these photosynthetic ICMs were transformed into respiratory cristae. So, well, I think this is a question that I would like to answer for my PhD. It is, well, did mitochondria evolve from a free-living purple bacterium that is a photosynthetic alpha puribacterium or a rickettsial endosymbiont? So I'm basically tackling this question from two very different perspectives. One is from the angle of the evolution of the internal architecture of mitochondria, and the other one would be phylogenomics by sequencing more genomes and then do more rigorous phylogenetic analysis. And well, that's pretty much everything I had to tell you today. Thank you for your attention, and I'm looking forward to answer some questions.
Well, thank you very much, Sergio. That was fantastic. I have to say, personally, that was one of the more understandable biology talks as a non-biologist that I was able to much more than I expected.
Oh, thank you.
Question? Yes, open the floor for questions.
There's
Yes.
a bit of feedback somewhere. I'm not sure. Anyways. Sergio, hi. Thank you very much for your talk. I really enjoyed it. I'm not a biologist either, and it was I was surprised by how much I was able to follow. So good job. If the proto-mitochondria was a complex alpha proteobacteria, what was the environmental pressure to cause it to endosymbiote with a proto-mitochondria?
Yes.
Like, if it was complex and happy, essentially, what caused it to invaginate and create the mitochondria?
Yeah. Right. So basically, in order to think about the reason why this alpha puribacterium became an endosymbiont, we also have to think about the nature of the host. How did the host benefit from the endosymbiont? So it turns out these complex alpha puribacteria are usually photosynthetic. So they perform photosynthesis to produce ATP, and they perform this photosynthesis in those ICMs. And different studies have actually shown that a lot of these bacteria secrete a lot of those photosynthetic products to the environment. So if you have a bacterium that is already secreting useful products to the environment, then any host can actually internalize that bacterium would benefit from those secretions, because those secretions can then be used for different processes. So that's what I think was the reason for the initial endosymbiosis, the secretion of photosynthetic products to the environment that the host could have used for their metabolism.
Thanks. Is that testable?
Well, every evolutionary hypothesis is very difficult to test, but we can basically accumulate indirect evidence and measure like how plausible each one of these alternatives is. And also, for example, doing phylogenetic analysis will shed some light on this, because if it turns out that the mitochondrial lineage actually branches with endosymbiotic Rickettsialis, I think we will have to discard my hypothesis that the ancestor was a complex bacterium, and we will have to think about how a more simplified bacterium established a relationship with a host. So there are different directions that we can take in order to try to test this hypothesis. But we will never be able to definitely say what happened.
There's a question in the chat room.
Okay. I will wait for my turn after the question here. Do you see the question on the? Okay, I'll read the question for you, Sergio. Do purple non-sulfur alpha proteobacteria typically respire oxygen as a phenotype within the clade?
Yes, that's true. Purple bacteria have basically the same respiratory chain that mitochondria have. So the respiratory chain of mitochondria is just a reduced version of the respiratory chain that you find in purple bacteria. So some reduction had to happen, but basically it is just a subset. So it's not difficult to imagine how this could have happened. So in terms of respiration, alpha proteobacteria have everything that is needed for optimal respiration.
Sergio, I am interested in hearing your thoughts about potentially reconstructing the ancient Mike 60 gene and engineering the bacteria with that.
Yeah.
Would you be interested?
Yes, I would be interested. The thing is that MYC60 is a very divergent protein, and I'm a little bit afraid of how robust the inference of the ancestral sequence will be. But what I think we could try is to reconstruct the sequence and then, or at least reconstruct the eukaryotic sequence and then try to insert it into alpha proteobacteria to see what it would do with alpha proteobacteria.
Yeah. Do you have like sequences for that? Because I think you did some phylogenetics studies yourself.
I do have a broad sample of sequences from different eukaryotes and also alpha proteobacteria.
Yeah, we should be able to do that fairly straightforward, actually. If but we can chat afterwards. But if you're interested in something like that, at least I think it would be valuable just to see how the ancestral one looks like, sequence-wise.
Yeah, definitely.
Yeah?
Yes, I think we should talk about this.
Do you have the structure information for Mike 60?
No, there are no structures for these proteins, unfortunately.
Is there a specific reason for that? Is it difficult to crystallize, or?
I don't know. I have no idea. I can ask the cell biologists who are directly studying the protein complex in mitochondria to see if they have any plans of getting the structure of this protein.
Yeah, it would be interesting. Like, what would be the first thing you would look at once you have the ancient sequence for this gene, just by looking at the sequence?
Yeah, I think it would be very, very useful to have the structure of the protein.
Transmembrane proteins are hard to crystallize. That's true.
Yeah, although like this protein has only one transmembrane segment. It is not a polytopic protein, so maybe that would make it a little bit easier. I'm not sure.
Yeah, I think we should definitely explore. It seems very suitable for that, and like I wonder what you would see if you engineer your bacteria with an ancestral version. Either eukaryotic. Yeah. I think Sergio has experience with the bacterial, like engineering the alpha proteobacteria. There's a question here that says you guys should consider either a eukaryotic or a bacterial host for your reconstructive approach. I will read you another question. How can you discount convergent evolution? That's a question for you, Sergio.
That's a very good question. Convergent evolution in the sense that both alpha proteobacteria and eukaryotes have achieved the same solution of using the same protein for creating membrane invaginations. Is that what you mean, Pak? Okay, so the protein in eukaryotes is clearly homologous to the protein in alpha proteobacteria. So, well, there's a direct genealogical connection between the two proteins. But in terms of function, I think it would be very difficult to discount convergent evolution, because actually I think that the protein might be performing a more general role in envelope biogenesis, and that the protein has been recruited independently in some lineages and also in eukaryotes for helping the development of cristae and ICMs. But it has been recruited precisely because the protein is very useful in connecting the two different membranes. So, well, yeah, I think it's very difficult to discount the idea that the function of the protein has been independently recruited for the same purpose, and that would be convergent evolution. Basically, but what I want to know is that whether the protein can have the same function in the two very distant groups, that is mitochondria and alpha proteobacteria. And if that is true, I think that would be evidence for the idea that cristae could have evolved from ICMs, although I don't think it would be definitive.
There's another question here. Do the mycos family of proteins have other functions in bacteria? Also, are they found across other clades as a result of horizontal gene transfer?
Yeah, surprisingly, it looks like, well, there's only one mycos protein that is found in alpha proteobacteria. And we don't know the function of that protein in alpha proteobacteria. That is what I'm investigating right now in order to test the homology hypothesis. And surprisingly, it looks like the protein has not been transferred to other groups of bacteria, because when I do searches against UniProt, for example, all the results I get is from alpha proteobacteria, even though we have more than 3,000 or 4,000 genomes for all eubacteria. So it looks like the protein has not been transferred horizontally to other bacterial clades, which it's difficult to imagine, but I can think of different examples of different proteins that have not been transferred horizontally. I don't know what the reason for this is. It might be that the protein has many interaction partners, so that makes the protein less prone to be transferred. But it looks like the protein has not been transferred to other clades. I think there's not. Okay. Betul?
Oh, no, I was wondering, there's a follow-up question asking, this is interesting, can these proteins be used to estimate the timing of the emergence of the eukaryotic cell, or do you think it is too narrow of a slice of information? I love this question. I can't wait to hear your answer.
I think it is, we don't have a lot of information with this protein, actually, in order to try to estimate the timing of the emergence of eukaryotes. We have to use as much as data available as we can. And very recently, actually, a paper in Nature appeared that addressed this question. And if you give me a second, I will find it and I will send. Basically, the authors investigated all the proteins of eukaryotes. I mean, all that are present in the genomes of eukaryotes, and they did chronograms, that is, dated phylogenetic trees, and they classified these proteins according to how old each one of these proteins was. And they found that the mitochondrial proteins relative to endomembrane proteins and nucleus proteins are the most recent proteins. So it turns like, according to this analysis, it looks like mitochondria actually came last from all the other eukaryotic innovations that happened during the emergence of eukaryotes.
Wow, that's interesting. The person asking says that this conflicts with Nick, I believe he's referring to Nick Butterfly, his Lane's theory. Nick Lane's theory.
Yes. Yes, that's true.
Could you maybe in a couple sentences tell us about what theory is that?
Okay,
It's
so
just exactly.
in terms of the origin of eukaryotes, there are basically two competing hypotheses. Well, there are several, but these are the two main competing hypotheses. One says that the host that internalized the mitochondrial ancestor was a host that had the capability of doing phagocytosis, which means that the host probably had some primitive cytoskeleton and probably some primitive endomembrane system. The second set of hypotheses basically state that the host was an archaeon that had no internal complexity. It was a simple prokaryote. So one prokaryote internalized another prokaryote. And the hypothesis of Bill Martin and Nick Lane states that the host had to be a simple prokaryote, an archaeon that had no endomembrane system or no cytoskeleton. And the reason for that is that mitochondria was the source of energy that allowed all the other eukaryotic features to evolve. I mean, they argue that the endomembrane system and the cytoskeleton cannot evolve if you don't have basically an energy factory inside your cell that will sustain the expression of all those proteins, and that would be the mitochondria. So in their hypothesis, mitochondria came first, and that increased the production of energy, and that subsequently allowed the evolution of a lot of new protein families that would uh, establish the endomembrane system and the cytoskeleton.
I see. That's wonderful, and it's a very if there's no other question, that also marks it as a very big ending. I believe you're going to be exploring these different ideas during your PhD studies and even further. And is there any other question from the audience?
I just shared the.
Oh, okay, great. Thank you. Thank you for sharing that. And if Jacob is here, I will leave it to Jacob to finalize. But thank you for your time, Sergio. And I'm sure we will hear from you again. Your presentation was wonderful and your slides were very explanatory. It's very good job. You managed to talk about molecular biology without a single molecule picture. Thank you for that. Yeah, and thanks everyone for coming. Yeah. Yeah, come to BlueSciCon.
Absolutely. I think you'd be a great fit at BlueSciCon, Sergio. It was very much in line with the kind of see there.
Okay, thank you very much for the invitation again, and I hope to join you for future sessions.
Thank you. Thank you, Sergio.
Thank you.